Hey everybody, so this video is for week 4 and lab group A, unless you are in Mr. Newman's class, in which case you are on week 3 this week. All the due dates for your specific section are in your lab schedule in Blackboard, and any specific questions about those due dates should be addressed to your lab instructors directly. Okay, so Group B this week is working on an at-home experiment based on the results that you obtained in Experiment 3. So we named it Experiment 3B. You're going to learn how to use Excel or Google Sheets in this week's work. And it's an essential tool in all of science. It doesn't matter which kind of science you intend to pursue. We use spreadsheet programs all over the place for a variety of reasons. So first off, the links in the beginning of the procedure are going to help you either obtain Excel or sign up for Google Sheets, one or the other. If you haven't got those already, you can get all of the MS Office programs for free as an MVCC student. So I recommend that anyway. But if you're more comfortable with Google Sheets, you could also use that. I have also linked here some video tutorials that will walk you through all of the different aspects of our experiment, our project today in Excel. Well, not today, this week. So if you're using Excel, you'll click the first link for the video tutorials about Excel. If for 2016, if you have Excel 365, which I think is the free one you get as a student, I like this one better. It has more features anyway. You can click on this link for the tutorials for that version of Excel. If you're using Google Sheets, you can click on this link for a whole bunch of video tutorials about how to do anything you might need to do, like resizing cells or columns, how to type in formulas. All the details of the sort of mechanics of the experiment can be found in any of these 3 links here, depending which version you have. The other thing to look at before you get started is to read the page, maybe it's 2 pages, about graphing requirements. And so the picture shown there is kind of an indication of how your Excel graphs should look anytime you submit them for scientific purposes. So for this entire year of general chemistry. All right, and so understanding what you're shooting for is important before you get started, so look at that. Then you're going to sit down with the procedure and you're going to use the data provided to you by your instructor for Part A. So this is a very simple temperature calibration graph that you're going to make. This is how we come up with the equations to convert Celsius and Fahrenheit. You're going to do one for millimeters and Celsius as well. And this will give you kind of an introduction into relatively simple graphing techniques in Excel. Then, by the way, navigating through Google's documents, it's highly recommended you use the table of contents on the left. If you don't see that, it's right here. So make sure you can use that outline to help you navigate. So Part B is where you're going to create the graph based on the data from Part A. Once again, the actual numbers from Part A are going to come from your lab instructor in your email, but the creating the graph is based on the work you did in Part A. Once you have a graph, you can create a trend line, which is like an average of all of the data. It shows kind of the pattern of the data, the trend. It does not mean we're playing connect the dots with our data, okay? A trend line is either a very straight line between all of the data points, or it could be a smooth fit curve, depending on the kind of data you have. In this case, we're only going to do straight trend lines. And so don't show connect the dots on your graph. That's the wrong design. That's useful for other kinds of data, but not scientific, not chemical data. Okay, so we're going to do a trend line. These are the instructions for that. And then you're going to do kind of the same process. You're going to have data table, you're going to have a graph, you're going to generate a trend line, this time with a little less detail. So we're giving you the temperatures to use, but you're going to kind of go through the same ABC process for new data to create a second graph which converts 2 familiar units. I also have included this kind of FAQ built right into this document. So if as you're working, you get confused about how to do something that's described in the procedure, you can click on the How Do I link. And it's probably explained there. Primarily in terms of how you'll find things in Excel. Okay. So then after you have finished that part 1 project, you're going to tackle part 2, which is based on your data from experiment 3 last week. And so you're going to use the masses and the volume that you measured for your salt solutions to create a standardization curve, ultimately. All right. And so first we have to make the data tables correctly. You're going to have to do some calculations in Excel. I do not want you to do them by hand. The whole point is to learn to use Excel to do your work in a simpler and more repeatable fashion. So you can type the calculation in one time, drag it all the way down the column, and it will do the calculation for you over and over again. It's a very, very useful tool to have in science. So that's what you're learning. And you are going to be graded on the formulas. Okay, you're submitting your actual document, either your sheet or your Excel workbook, so that your instructor can see the formulas you typed in. Okay, so the instructions for the formulas are in the procedure. Follow it. Don't deviate. Even if you think you know Excel, you probably don't know it the way that science uses it. And it's important to line everything up in the right cells so when you get to the calculation parts, everything makes sense. Okay. So then you're gonna create your data table, and then after that you will create another graph. Okay, and this one is going to be used to determine the concentration of your unknown salt solution that you tested in lab. All right, so the unknown is not included in this in the initial data table. As part of your graph. You can put it in the data table, but when you go to highlight for your graph, don't use those points. So it'll just be 0% to 20% and everything in between. You have to know the concentration of the material in order to make a standardization graph. So the unknown won't go on that table directly. Instead, it gives you instructions for how to show using shapes drawn on the spreadsheet where your unknown happens to occur. Okay, so this is called graphical analysis, and so it's used anytime we have a graph and we have one variable we know about and we want to find the other one. You basically just draw a line up to your trend and then draw a line over, and it'll show you the answer to the question of like what percentage your salt solution unknown was. The other approach to that is you can also use the equation of the line to find the same thing. Be careful to replace y and x with the actual variables. So you need to put the percentage concentration and the density in the right place in that equation. Okay. So this report is actually the same report as experiment 3. So really the work you're doing here is part of the calculations in experiment 3. And so they're all going to be included together in one final report, so make sure to check your lab schedule for those due dates. Okay, so what you're turning in for this experiment this week is there's a pre-lab at the end. You're going to do that in your lab notebook. And you're also going to submit a copy of your actual spreadsheet file. So if it's a Google link, you can give the link to your instructor, or if it's an Excel sheet, you're going to upload the whole workbook. All of your graphs need to be in one spreadsheet program. Don't make them in separate programs. You can add new pages to the same workbook by clicking at the bottom. Okay, so everything goes in one file, and you're going to upload the one file along with your pre-lab for grading this week. In addition, you have some other stuff going on this week, so it's important to keep track of the lab schedule. So for Group B, you're going to want to slide, and this is just for my lab, so you need to look at your exact due dates, okay? But I think every instructor has them in Blackboard, and so what you want to do is go look at that. And so this week we're doing experiment 3B. The report for that is not due until 9/22, but your pre-lab and your Excel file in my class need to be turned in. Whoops, wrong one. That's the pre-lab for this one. I'll have to add that. I'll put a date on that, so check that out. But the other thing that we do have coming up this week for most of our sections is you're going to have the separation of a mixture lab report that's due. So in order to know how to do that, you need to actually look at the procedure, and it lays out all the components for the report. Okay, so right here under the report heading, you'll find a description of everything that needs to be included. When you are scanning in your handwritten work, please make sure you're following tech tip number 3. It all needs to be one file, not separate images, and it needs to be really easy to see because computers can make it difficult to begin with. So you need to do everything you can to crop it, have good lighting, all those parameters. So I show you that in this tech tip. Okay, and so what you want to do is cover each of these sections about this lab report, okay? And the calculation section is really just one calculation. If you did not obtain a melting point, then you're not going to be able to do that calculation. You'll have to come back to that once you do get a melting point of your material. You can discuss that situation with your instructor if you have concerns, okay? And there's not going to be a discussion and conclusion section for this report, although most of the other sections are there. So instead, you're going to just answer this series of questions by typing your answers out. Okay, so lab reports are formally typed, size 12 font, 1-inch margins. All of the standard requirements for writing are going to apply here. And remember that you can check out our introduction packet for further details about what goes into each section of a lab report. Okay, so if you ever forget where to find those items, they're also available right on the lab schedule that everybody has. It's right here, Introduction to Gen Chem Lab. And of course, again, just to point this out, all of your procedures are also located there. These blue links are the procedures. Lastly, you can find the same information about the procedure links by going to your group folder. The black links are where you are going to submit things. So like this week, your separation of a mixture report is due. So you'll click the black one and you'll submit the final report by whatever due date your lab instructor has established. Probably it's this week. Okay, and so then the blue link here, so the black link is for submission of assignments. The blue link is the procedure. So if you need to go back and refer to that again, it's there for you. Okay. So if you were not able to pick up your kit to do the separation of a mixture experiment in week 1, this is the week for the makeup kit to be picked up. So if you haven't done it, obviously you can't turn in the lab report this week. You do need to pick up the kit and go work through the procedure. The biggest tip I can give you from the last 2 weeks of people working on it is that you really need to look up those properties that are outlined in the beginning of the procedure and use them as a way to decide how to approach the experiment. Also, I would tell you it's a better idea to mix things together in the beaker or a cup first and then filter it rather than putting the solid on the filter and pouring the liquid through. Our filters work faster than coffee filters do, and so they don't trap the liquid long enough to dissolve things that are going to dissolve. So you want to use a little bit of a different tactic than we would usually use for coffee. And that's very common in science. We want to mix them first, then pour through the filter. If you need to pick up your separation of a mixture lab kit, you can do so Monday from 8 a.m. to 9 p.m., Tuesday from 5:30 to 6:30 p.m., Wednesday 8 a.m. to 9 p.m., although not if you have class, and Thursday 12 to 1. If those times don't work for you, please email your lab instructor and Kate so that you can make alternative arrangements. Okay, I think that's all that's going on in lab this week. As always, if you need help, reach out to me or your lab instructors. We are here to help you. Have a good week.